Hello everyone. This video is meant to review objective number 2 for your RNA test. This objective would have you describe the process of translation. So let's begin with an overview of translation first. This is a poster you should recognize from the classroom. Translation takes place in the cytoplasm of the cell because that's where the ribosomes are located, and the entire purpose of translation is to build polypeptide chains based on DNA's instructions. Now those polypeptide chains are meant to later fold into the proteins needed for your particular traits. So if we look at this diagram, you'll clearly see that there are 3 types of RNA that are in place during the translation process. The least obvious would be the RNA that forms the ribosome. And remember, ribosomes are made of 2 subunits of ribosomal RNA. This lower half would be the smaller subunit, and this upper half would be the upper subunit, which is a little bit bigger. The ribosome, as you remember, are the protein factories of the cell, but they can't do their job unless they have instructions from DNA. However, ribosomes cannot read the DNA language, and the DNA is locked away in the nucleus. So instead, the DNA will send its messenger. This blue strip represents messenger RNA that was transcribed in the nucleus of the cell. Basically, it is carrying DNA's instructions to the ribosome. However, it's carried in a readable form, because the ribosomes can read RNA that can't read DNA language. Also in the picture, you'll see transfer RNA. And transfer RNA's job is to carry specific amino acids to the ribosome. So the ribosome will receive the messenger RNA and begin reading it 1 codon at a time. And if you use your chart, GCC stands for a particular amino acid. That's this pink dot right over here. To get the amino acid to the ribosome, we needed the transfer RNA that can temporarily bind with the messenger RNA while in the ribosome. The next codon is AUG, which stands for methionine. This would be the methionine amino acid, and it is delivered to the ribosome by this transfer RNA. Then you have UUC, which is a 3rd amino acid delivered by this transfer RNA, and here's a green one coming down based on this code right over here. So messenger RNA would be the readable instructions that the ribosome can then translate. The ribosome itself is made of RNA as well. And then the transporter of amino acids would be the tRNA. In order for transfer RNA to drop off its amino acid, it has to form a temporary bond with the messenger RNA while in the ribosome. So you look very closely, the codon and the anticodon of transfer RNA have to be complementary. These two will form a temporary hydrogen bond in order to drop off the amino acid. Once the amino acid is dropped off and links with the next amino acid in line to form that polypeptide chain, then the transfer RNA can leave empty-handed. Eventually, the polypeptide chain forms and we'll reach a stop codon, which means let's not add any more amino acids. This polypeptide is done. Then the polypeptide can fold into its proper protein shape to give you the traits that you need. This is a codon chart to be used with messenger RNA only. And as you remember, messenger RNA would be DNA's readable instructions. To read this chart, you need a codon. Each codon is an instruction for a particular amino acid. For example, here's a codon, AGC, which would be found on messenger RNA and would be read by the ribosomes. So to use this chart, you need to start with the very first nucleotide in the codon, which is adenine. So divide your chart down to find just the section for adenine. Then go to your next nucleotide in row, which is guanine. In the adenine quadrant, find the guanine section. And then go to your 3rd nucleotide, and in that section, find the C line, which tells you which amino acid you're talking about. So AGC stands for the amino acid serine. So when the ribosome reads AGC, it's going to call for a specific transfer RNA to bring serine down so it can bind it to the polypeptide chain. Now, if you look closely at this codon chart, you'll see that we've got a few things going on. Most of the codons are coding for particular amino acids. However, we have some unique ones as well. Now, as you remember, AUG stands for methionine. That's a very particular amino acid, but it also serves as the start codon for any polypeptide chain. Now, if you look over here, you'll see that we have 3 specific codons which don't code for a start or any particular amino acid. In fact, they code for a stop codon, which means stop producing any more amino acids. We are done with our chain. So you have UAA is a stop codon, UAG is another stop codon, and a third one is UGA. All 3 of those mean please do not add any more amino acids. This polypeptide is done. Now let's show you the process in action. Always begin with the master molecule DNA, which is locked in the nucleus. Now, DNA has 2 sides, right? But we're only focusing on the active side of DNA, which is this one over here. We're going to disregard the inactive side entirely. This active side will serve as our template for building our messenger RNA. So let's show you the process of transcription. We're going to take our active side of DNA to build RNA. Now, if we know that TAC is the DNA instructions, then we need to find the messenger RNA complement to TAC, which would be AUG. Thymine is complementary to adenine, adenine is complementary to uracil, cytosine is complementary to guanine. Remember, RNA does not carry any thymines, so in place of it, we'll put our uracil. The next DNA code, GGG, will be transcribed into CCC on RNA. Then you have TGT, which gets transcribed into ACA. Then you have CAA, which gets transcribed into GUU. Then you have ATT, which gets transcribed into UAA. Now remember, the DNA itself does not get altered. It stays exactly the way it is. We're only using the active side to assemble these RNA nucleotides in a very specific order. Now, as you remember, each RNA codon, like AUG and CCC, etc., stands for a particular amino acid. So now we need to do the process of translation. Using your codon chart, you remember that AUG stands for a particular amino acid, and the goal is to build a polypeptide chain. So let's find out what AUG stands for. Methionine. CCC on the codon chart stands for proline. ACA stands for threonine. GUU stands for valine. And UAA is a stop codon. No amino acids here. So if we put those particular amino acids into a sequence, methionine, proline, threonine, valine, we have our polypeptide chain. The polypeptide chain can then fold up into the final protein structure, which we then use to determine your traits. For example, maybe this protein would allow you to have green eyes. I hope this helps. Happy studying.